Dr. Hashim know that I'm that SI leader for that class. And her final is going to be 75% cumulative material and 25% material from this past module. So because you guys have had all of that information in class in the past week or so, I didn't focus this review on the chapter 13 and 14.1 section. This review is more for the cumulative side because you haven't seen that in a while, and that's what the main focus of this is. That being said, if you have any questions, hold them till the end and we'll go over them. Other than that, if you have any other general questions for me now, anybody? You can go ahead and ask them now. Okay, so we'll get started. So, first things first. Biochemistry, study of life, that's all this is about. I'm going back to the very basics. So starting in like chapter 1, all of that stuff that we talked about. We've got self-replicating biopolymers, nucleic acids, proteins, carbohydrates. All those are polymers. They're going to be put together from basic monomeric units. And self-replicating, that's how we got to where we are now. That's at least the current theory. It's all guided by this central dogma, and this is how in biochemistry we know where we're going. So we start with DNA, and then from DNA we make RNA, and from RNA we make proteins, and from proteins we make pretty much everything else. And that's why we focus so hard on proteins and all of these like macromolecule synthesis, is because we need them. Well, life wouldn't exist without them, which is pretty cool, at least in my book. I don't know. That's funny, you guys can laugh and stuff. Thanks. Gotcha. All right, so the next start in the beginning is thermodynamics. We talked about delta G and how that relates. So we've got free energy is equal to the difference of enthalpy minus enthalpy minus entropy times temperature. How many people didn't remember that one? Okay, good. I'm really hoping that you guys a lot remember these basics because they're going to be applied in the future. So we have gives free energy. It's going to become really important when we start talking about equilibrium constants because we've got this other delta G equation. Delta G equals G standard plus the R constant times temperature times the natural log of the equilibrium constant. Where have we seen that before? Anybody? There's only like 8 of you, so I want you guys to like talk to me and stuff. Where do we use this delta G equation? Well, just where do we apply them? Like what have we seen in biochemistry in the past semester? I'm like calling on your memory here. Of what? Okay, so biomolecules and their energy transformations. Did we ever talk about it with like proteins and stuff maybe? Sound familiar? Catabolism? Right. Metabolism and all that stuff. So we've got metabolism and all of these enzyme kinetics. They come back to this delta G equation with the RT, L, and K because we have to deal with the equilibrium concentrations, and that applies when we talk about Michaelis-Menten, which we'll get into in a bit. Okay. So we talked about the water stuff in the beginning. Universal solvent. Who remembers the Henderson-Hasselbalch equation? It's not a trick question. It's right there on the board. Anybody want to tell me? Someone shout it out. Anybody? Anybody? Come on, you guys. All right. Literally on the board, Henderson-Hasselbalch, pH equals pKa plus the log of the base over the acid. That is hopefully something you remember, because it becomes really important when we talk about a lot of the ways that enzymes and stuff work. We spent like 3 weeks on this, I think, in Dr. Hashim's lecture. So I hope that that stuck with you. Does anybody have any questions about the Henderson-Hasselbalch equation and when it was applied? Because I think I have a practice example that I'm going to make you do like right now. Do what? Did we? Can you tell me? This concept applied later on when we talked about enzyme catalysis. We talked about like how the concentrations all relate and how all the protein kinetics is controlled by the pH. What changes when we change the pH? Charges of R groups? Okay, that's one thing. What else changes? Okay, so we're changing the concentration of H+. That's going to affect how our proteins interact. Does changing the pH affect the ability of the proteins to work anymore? Yes. What is that? What can that cause? Big fancy word starts with a D. Anybody? Denaturation. Yes, thank you. So we have to be careful with pH because our enzymes and our proteins are very specific to the conditions that they're in. Everybody cool? Okay. I'm calling back on basic fundamental stuff because the exam is pretty close, and so hopefully you guys have studied at least a little bit by now. This review PowerPoint is going to be posted online too, so you guys will have access to that after tonight. Practice question, yay! Okay, so I don't want to, because we have so much information to go through in such a short time, I don't want to like work it out step by step, but someone give me like a brief idea of how we would approach this problem. It says a solution is made by mixing 50 mils of the 2.0 molar potassium phosphate and the 25 milliliters of 2.0 molar, I don't remember the difference and how to name them, but the other version, the solution was diluted to a final volume of 200 milliliters. What's the final pH of the solution, and is it an effective buffer? How do we approach this problem? Anybody? Yeah? Okay. Okay, awesome. So his suggestion was to start with the Henderson-Hasselbalch equation and figure out our concentrations so we can plug those in. Okay, so if we find the concentration of the base and we find the concentration of the acid, what's the other part of the Henderson-Hasselbalch equation we need to fill in to get the answer to this problem? pKa. Okay, we have 3 pKa's listed on the board. Which one do we use? Somebody else. Anybody? Third one? Kazili section, you're allowed to participate as well. You guys can like talk to me and stuff. It's totally allowed. So Stephanie back there says the third one, why? That's the one for the final solution? You guys tell me. What are the two components, or what's the acid that we're dealing with here? Okay, so H2PO4, that's the acid component of this buffer system. So which one of those pKa's is representative of the proton that's going to come off of H2PO4? pKa1? I gotta vote for 1, I gotta vote for 3. Anybody vote for 2, just for fun argument's sake? So I'm glad we're going over this topic because I feel like there's a lot of confusion here because it's actually pKa number 2. So when we start with phosphoric acid is a triprotic acid, right? It's got 3 protons in the beginning. H3PO4 is the fully protonated version, right? This buffer system doesn't use H3PO4. It's using H2PO4 and HPO4. So we have to then lose the first proton. That took care of pKa1. Now we've got 2 protons left, and that's our acid in this case. So pKa2 is going to be representative of the proton that's going to come off to form HPO4. Does that make sense? Does that clarify that for you guys? Right. It's kind of the opposite. It's the pKa for the one that's about to come off, not for after. Okay. So if pKa3 is for the monoprotonated form, and pKa2 is for the diprotonated form, pKa1 is for the triprotonated form. Everybody cool with that? Knowing that information, could you solve this problem? Because I think you could. Okay, cool. We're going to move on because stuff. I have like 50 slides here in like 2 hours. So. Proteins. This is like the big part of biochem. Hopefully you guys aren't sick of talking about proteins yet because they never go away, right? So what are proteins made of? Anybody? Amino acids. Hopefully you know that by now at least. Like, come on, you guys. hopefully also you have your amino acids memorized. I know for sure Dr. Hashim's class should, because you guys had like 4 quizzes on remembering amino acids way back when in the beginning of the semester. They still exist and you still have to know them, but your exam is multiple choice, and so it's mostly just going to be a recognition game at this point. So don't get too bogged down if you don't know them exactly by heart right now, as long as you can recognize which ones are which. So we have the 20 amino acids. What are the different structure types for proteins? Yeah. Any level. What are the 4 levels is my actual question. Primary, secondary, tertiary, quaternary. Again, not a trick question. 1, 2, 3, 4. Super simple. Biochem's really easy, you guys. So we have primary structures. What are primary structures made of? Amino acids. And how are the amino acids held together for primary structures? Peptide bonds, right. Linearly. Awesome. Perfect. Okay, so we have our linear primary structure held together by peptide bonds or whatever. Pause for the cause. Quick question. What functional group that we learned in organic chemistry is a peptide bond? Amide or amino? Which one? Amide? Amide, yeah. Carbonyl, and it's still an amide. It's still the same functional groups. I had some confusion over that with some other people I was talking to that weren't in your class, but that's important to remember. Yeah? Right. Exactly. So when you have, what Ian said is when you have a protein together, the amino acids are held together by the peptide bonds, and that peptide bond is forming the amide functional group. But when they're not conjoined, the separate amino acids just have the amino group and then the carboxylic acid group. Thanks for bringing that. That's important too. So we have the amino acids in the primary structure. How do we get to secondary structure? Hydrogen bonding. Yes, thank you. So we have hydrogen bonds, and what are our 2 main kinds of secondary structures? Alpha helix and beta sheets. Awesome. What's your name real quick? Alex. Okay, I know a lot of you, but I'm working on it. So we've got alpha helixes and beta sheets, and they work together. How many different types of beta sheets do we have? 2. Yes, 2. What are they? Parallel and anti-parallel, right. So we've got parallel, we've got anti-parallel, we've got the alpha helixes, and how are they held together? Hydrogen bonds. Did I ask that one already? Oh, I forgot. Okay. So, of the backbone. Yes, that's the important part. That was the next part I was getting to. So we've got hydrogen bonding between the NH group of the backbone and the carbonyl of the backbone. Where do we not have hydrogen bonding in our secondary structures yet? When we're specifically considering the secondary structure. Right, between the R groups. When we're looking at the secondary structure, we're looking at specifically how the hydrogen bonds are holding the backbones together, not so much where the side chains are involved. When we're talking about the location of the side chains, what structures are we talking about? Tertiary, right. So the tertiary form is when we consider where the side chains are in relation to each other and how all of the different alpha helixes and beta sheets fold together to form little subunits. And when we talk about quaternary structure, what are we talking about? Multipeptide chains, right. We've got subunits that are coming together. Right, more than one polypeptide is a good way to put that. So we've got different subunits of tertiary structures coming together to form our full globular protein. Like hemoglobin, exactly. I was just about to ask for an example. Thank you very much. So we've got hemoglobin's got a bunch of different subunits in it. We've got 4 different polypeptide chains that are all folded into their respective tertiary structures that are then held together for protein. Make sense? Everybody cool? I like that model. That was beautiful. I love it. Okay. Does anybody have any questions so far? I'm going to go with no. Cool. So the first step of figuring out how proteins work is getting them isolated, right? We've got to know what we're looking at before we can try to figure out, or we've got to know the specific components of the solution we're looking at before we can figure out how the proteins are sequenced. So does anybody want to list a couple of other types of chromatography? I've got gel filtration up here. What are some other ones we saw earlier in the semester? Ion exchange, what did you say? ELISA's a good one, yep. Any other purification methods we remember? There were like a billion of them. Which one? The blotting patterns, yep. The western blot and the southern blot and the northern blot. There is no eastern blot, ha 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 ha, funny. Anything else? What are some other isolation or purification methods. SDS page, okay, that's another one. We've got affinity, we've got ion exchange. Now we have affinity because I just gave it to you. We've got gel filtration, all these other ones. Anybody think of any others? I already forgot which ones we said too, so. Yeah, so that's a good question. So how is this gel filtration system, how is that different from gel electrophoresis? Right. So with gel filtration, we're looking more at the gravity being using gravity to pull the solution through the column. Whereas with gel electrophoresis, we've got a bunch of charged molecules and fragments, I guess, that get pulled through at different speeds because of their charges. So is everybody cool on that part? Okay. So in gel filtration, what comes out first? Big or small? Small? Big? I've got some votes for big, I've got some votes for small. Big? Okay. Why is it big? Why do the big ones come out first? Dimitri, say that louder. Exactly. So the small ones are going to get stuck. We've got these gel beads that are inside our column, and they've got these pores in them that kind of just like the little bitty ones, they get stuck. The big ones go around those pores, doesn't matter. They don't fit, so they're just going to pass right through the column. So they're not going to be slowed down as much. They're going to come out of the column first. We can collect that one. We can move on, and then we can wash out our small molecules and get those in our second fraction when we do our chromatography. Make sense? Okay. So I think I've got some other types up here. Ion exchange. What comes out first in a cation exchange column? Negatively charged comes out first? Positively charged comes out first? Positively charged? I have different votes here. So let's talk about why this works. So does anybody remember what CM and DEAE stand for? No. So it's okay if you don't. Close. Diethyl aminoethyl. So the DEAE is diethyl aminoethyl. And if you happen to know the structure of it, then cool. If not, then just remember that that's the positively charged thing that's attached to our matrix. But that, as it's listed here, is the anion exchange column. So what happens is you've got that positively charged matrix, and we put our solution through. What gets stuck to the positive? Negative. Right, thank you. So if the negative ones get stuck to the column, the positive ones are going to get repelled first. So in a cation exchange column, you have the opposite. So you have carboxymethyl, which is the negatively charged matrix. What's going to stick to the negatively charged first? Positive. So what's going to come out of the column first? Right. So in a cation exchange column, the negative comes out first. Make sense? Cool. All right. If you are thinking about that on the exam and you feel like you're getting tripped up, just remember the structures you're looking at. Dimitri. Okay. So if we have the cation exchange column has a carboxymethyl matrix attached. Carboxymethyl being an anion, what's going to stick to the anions first? The cations. So we're going to exchange whatever was stuck to our carboxymethyl in the first for the cation that's in the solution. And that's going to get stuck to our column. So the first thing to come out is going to be the anions, right? Because they're going to get repelled and pushed through the column a lot faster than the positive ones are going to get stuck. And it's so whatever the way that I like to remember this is whatever the name of the column is, comes out last. So for the cation exchange column, that comes out last. For size exclusion, that's kind of a harder one to remember, but it depends on what your column is. So, what? Right, it's going to grab that cation out of solution, and it's going to hold it, and it's going to take longer for it to get out of the column for the cation exchange. And I remember it by what the matrix is doing, because I remember positive attracts negative a lot better than I remember all of these other little minor details. But that's just me. Okay, so what about affinity? Let's say we have an affinity column and we're using a specific antibody and antigen combo. What's going to come out of the column last? Something with high affinity or something with low affinity? Low affinity is going to come out last? High affinities come out last. Right. So if the column really, really likes the material that it's looking for, it's going to hold on to it a lot longer, and so it's going to take longer for it to come out of the column. So for the affinity chromatography, whatever has high affinity comes out last, just so that we're on the same pattern with like cation exchange, cations come out last. High affinity comes out last. So SDS page. This is, I know when we talked about this first in the semester, it kind of got brought up a lot in SI, so I wanted to make sure we covered this one here. So how does SDS page work? Does anybody remember? Yeah, Ian, go ahead. Breaks hydrogen bonds? Okay, so if we're breaking hydrogen bonds in our proteins, what are we breaking up? Secondary structures, right? Because that's how they're held together. Okay, what else are we breaking up? Let's say we have a tertiary structure. How are those subunits held together? Disulfide bonds or bihydrogen bonds, or which one? Both? The tertiaries are held together by disulfide, that's right. So with our tertiary structures, we're going to have a lot of disulfide interactions. It's going to affect the folding. But then for the different subunits for the quaternary structure, a lot of that's going to be held together by hydrogen bonding, and so we can break that up with SDS page. So we're going to denature those proteins, and we're going to be able to fragment it. So now we've got, based on the size of the fragments, the ability to separate our proteins. If we're looking at this gel that's on the screen here, and let's say we went ahead and broke up all the disulfide bonds with the mercaptoethanol reagent, where are our largest fragments? At the top of the gel or at the bottom of the gel? Top, right? Exactly. The larger fragments are going to be closer to wherever you put them in. So if it's at the top or at the left or however your charge system is moving across. Why is that? Exactly. The smaller ones are going to go faster through the gel. Ideally, all of your fragments have the same charge, and so they're going to have the same velocity otherwise related to the charge gradient that's going across the gel. And so the only thing that's going to be separating them is size. And then we can do SDS page. Make sense? So what's SDS page good for then? Separating by mass, right? Is it better for isolating a protein in solution or identification of what that protein is? Or like sequencing, I mean. Isolating? Or sequencing? I've heard both of those. This one's actually better for sequencing because you get the exact sizes of your fragments together. So if you have certain bands in certain places, you can know that, okay, this band is, say, 100 kilodaltons, and this band is 30. I know I've got specific subunits of those sizes. And that can lead a lot more towards the identification process. Because we're destroying our proteins in the process of this, we're not going to be able to collect them back and reuse them without special reagents. Does anybody have any questions on the chromatography stuff so far? You're allowed to ask questions, I swear. Yes, that is a gel. Oh. It's a good question. How is a Western blot different from SDS page? Anybody? We've got antibodies linked to it. With Western blot, that's essentially it. You've got a lot of extra reagents involved so that you can actually collect just specifically your tagged protein fragments or whatever you're looking for, and you separate them out onto another matrix, and then you look at that matrix. And so the Western blot, which one? So there's Western, Northern, and Southern. Which one's used for which? Western's proteins. Which one's Southern? DNA. And which one's Northern? RNA. Sound good? Snowdrop, right, exactly. Okay. And then this is what we were talking about earlier with the ELISA. This is a very similar process. It's kind of like the Western blotting. You've got a matrix, you've got the antibodies on it, and then you put your solution on it and they get stuck. The ones that have affinity for the antibodies get stuck. And then you wash away everything else, and then what you're left with is your protein that you're looking for. And then you can use that protein in a chemical reaction and do stoichiometry, figure out how much you have. Do you guys remember how this all works? I I know Dr. Hashim, I don't think she focused a lot on it in the beginning, so I wanted to bring it up here just in case, but I don't want to spend a lot of time on it. Yeah, essentially. So Ian was doing a lovely demonstration with his phone of how it works, but you've got the antibody, your substrate comes in, or your protein gets stuck to the antibody, and then you introduce your substrate, you get your product, and based on the concentration of substrate and product, you can figure out how much protein was there to begin with. And that's all the enzyme kinetic stuff that we'll talk about, I think, next or soon. Nope, still on this stuff. So we talked about STS page and figuring out fragmentations. What are some ways that we can fragment our proteins? I'll give you a hint, there's one on the board. CMBR, cyanogen bromide, yes, that's one. What are the other ways that we use to fragment proteins for sequencing? Which one? Sorry, say that one more time. Sanger sequencing. Is that one proteins? I don't remember the specific names of them. I have to go through the processes. What are the specific chemicals we use to fragment our proteins? There were at least 3 of them we had to remember for Hashim's class. Serine proteases, right. So we've got these serine proteases that are going to cleave our peptides. What are the main ones, at least the ones we talked about a lot in class? Trypsin, chymotrypsin, and which one? Mptopeptidase V8. Sound familiar? Edmond's reagent is related. So Edmond degradation is going to be the next step that we're going to talk about. So if we're using these serine proteases to fragment our proteins, where do those serine proteases fragment? Where does trypsin fragment? Anybody remember? Arginine and lysine, yes. Question? Yeah, kind of. So because the tumor captoethanol is used to reduce the disulfide bonds, so it's, in a way, it's fragmenting, but it's not breaking peptide bonds. So these specific ones, like the cyanogen bromide, which isn't really an enzyme, and then the serine proteases, those specifically cleave peptide bonds, and that's how we can separate the fragments. Right, for sequencing, you would want to split up the disulfide bonds in some cases. In other cases, I've seen problems where they fragmented them with the disulfide bonds still in, and then without them, so that they could see where the pieces, right. Okay, so once we have our fragments made, we're going to use trypsin, chymotrypsin, and endopeptidase V8, and maybe cyanogen bromide. So we said that trypsin cleaves at arginine and lysine. Where does chymotrypsin cleave? Right, so we've got the aromatic ones. That's how I remember it. The trypsin, or chymotrypsin cleaves all the aromatic ones. So tyrosine, tryptophan, and phenylalanine are all going to fit in that specific pocket for chymotrypsin, and then it's going to break the peptide bonds in the serine protease mechanism, which we're actually going to see later in this presentation. Okay, so we said trypsin is arginine and lysine, chymotrypsin is the aromatics. Where does endopeptidase V8 cleave? Elastase? Okay. I know she gave you guys 3 specific ones to remember, and I think you're right. I think it was elastase for Hashim's class. Sorry for Kazuli's section. I don't know which ones you're required to remember, if any. But for elastase, where does elastase cleave? You sure? We got 3 of 4. We had valine, alanine, glycine, and there's one more that's not threonine, it's serine, right. So SVAG, however you want to remember that. I could come up with some mnemonics, but I don't want to right now because we don't have a lot of time. So these cleaves, and those are the 3 serine proteases that Dr. Hashim wanted you guys to remember, at least for that exam. So once we cleave them, how does the sequencing work? Oh, I forgot one. Where does cyanide and bromide cleave? Methionine. It's right there on the board. You guys can talk to me, I swear. Okay. So now we sequence them. How does this sequencing thing work? I know we worked a lot of problems on this, and it gets really complicated, so I want to make sure if this comes up on your exam, you guys know how to do it. Okay, we have to use 2 different cleavage mechanisms. Right. So what Ian was saying is we have to have 2 different cleavage mechanisms so that we can have 2 different sets of fragments so that we can then compare the fragments and overlap them to get our original sequence, right? So we have to have like at least 2 protein molecules. Like we can't just do this with one. That would be a big problem. So we have 2 separate flasks. And in this one, let's say we use cyanogen bromide because it's on the board there. And so in the flask in my right hand, I've used cyanogen bromide, and I fragment them all at the methionine. And in this one, I use trypsin because it's also on the board right there. And I get my fragments. Then what can we do from there? Piece them together. Exactly. That's all you got to do. Right. So in the first flask with cyanogen bromide, we'd see that first 3 amino acid fragment that ends in methionine. But in the second one, our fragments wouldn't end in methionine. They'd end in either arginine or lysine. And so we'd have a longer fragment. And based on where the location of that first fragment was in our other flask, we can take those the sequenced fragments and put them together. Why do we do that in the first place? We have to sequence these fragments anyway. Why can't we just sequence the whole protein and be done with it? Has to be shorter than 100? Why? Don't know. Let's think about this from a logical perspective. Let's say we have a protein and it's got like 10,000 amino acids in it, and it's really, really big, and we want to sequence the whole thing. Is it going to be time/money efficient to sequence the whole thing with Egman degradation? Probably not. It's going to take forever. We're going to lose a lot of material in between every run of the reaction. It's going to be really inefficient, and we're going to want to hate ourselves after the end of it. So instead, we're going to cut this up into manageable bite-sized pieces that are like maybe like 10 or 20 fragments long. It's still going to take a little bit of time, but we're not going to lose nearly as much product, and we'll be able to sequence our fragments and then go from there rather than trying to get the whole thing set up. Cool? Okay. So now that I've mentioned it, what is Edman degradation? Anybody? Right. It cleaves off the N-terminal amino acid, just like we have on the board here. So we put in our Edman reagents, and we can take off the very last amino acid, and then from there we can look at, okay, this amino acid was alanine, and this one, the next one that came off was glycine, and then from there we can get our fragments. So if we know the N-terminal sequence, then we can just put it all together. We know our fragments. We can do the overlap. Edman degradation is kind of the glue that holds this whole sequencing process together. If we don't have this, we can't do anything at all, and that's a problem. I don't think, does anybody remember if she said like specifically which structures and reagents you have to remember for Hashim's exam? Okay. I know for the first exam that this was on, I think it was exam 2, she didn't make you guys know the reagents or anything like that. So don't like freak out too hard if you can't remember specifically that it's phenyl isothiocyanate that's used for Egman degradation. You can just remember that this is how this process works. That's probably going to be enough for you, hopefully. So just understand that you're cleaving off the N-terminal amino acid, and that's how you're sequencing your protein fragments. Okay. Who remembers this concept? Electrospray ionization? Yeah, I thought so. That's why I wanted to bring this one up. This one brought up a lot of problems in SI, and we didn't really talk about it for more than like a day in lecture. But they're very common questions, and I saw a lot of practice multiple choice when I was looking just like through the book and stuff in general about ESI. So I wanted to make sure we go over it. So what are we using electrospray ionization mass spec for with proteins? Okay, it gives us a charge to mass ratio. What can that tell us? Okay, Okay, so we can find out how many charged amino acids there are, right? So this is a great technique for identification because you're right, it narrows down the sequences of proteins that we could possibly be looking at. So for ESI, we get a spectra that looks kind of like the one that's shown on the board here, which this one is 20 charged amino acids in the sequence. But how does ESI work? Does anybody remember? Good thing we're going over it then. So for electrospray-ionization, the first part of it, obviously, is the electrospray part. We're going to spray our macromolecules into the air, but in a tube in the machine, not just like into the atmosphere. That wouldn't be useful. So we're going to spray these into a capillary tube in our instrument, and then we're going to ionize them. We're going to shoot a bunch of electrons at it and a bunch of energy at it until they all become the positively charged ions. When they're positively charged ions, they can be picked up by the mass analyzer, and we can get our mass to charge ratio. Instead of normal mass spec, which you guys might remember from Org 1 and 2, is specifically like making fragments that are charged. In this case, we're not fragmenting it. There might be a little bit of fragmentation, but it's not going to be nearly as profound as the peaks from just the charged whole protein. And so each one of these peaks represents the entire protein by itself. So how can we figure out, if we know the mass to charge ratio for all of these different peaks, how can we figure out the mass of our protein? Which one? Well, that's what we're looking for. So if we're looking for molecular weight, how can we use this chart to do it? Right. So the last peak on our spectrum, the highest one, is going to be the equivalent of our m plus 1 over 1 peak. I'm going to explain why that makes sense. So if we have, say, 5 amino acid residues in our chain that are able to become positively charged. So those are the arginine, the lysine. Are there any others? I think it's just and histidine sometimes, but they don't normally count it for ESI. So we got arginine and lysine are the two that we're looking for. Let's say we have 5 of those residues in our protein. We're going to only see 5 peaks on our spectra because we're only going to have a maximum possible charge, I guess, of 5. And it's totally random as to which macromolecules form which ions. So we're going to shoot energy at a cloud of proteins. And there's going to be, a billion something proteins just in our spray. We're going to ionize each one of them. And if it's random at which ones get ionized when, then we're going to see some that are 5+, some that are 4+, some that are 3+, some that are 2+, some that are 1+. And then some of them might not get charged, but we're not going to see that on our spectra because we can only see them if they have a charge to begin with. That's just how the mass analyzer works. And so that last peak in our spectra is going to have that +1 charge because if all of our molecules have the same mass and the only thing we change is the charge, then m plus 1 over 1 is one value, m plus 2 over 2 is a different number. Plug in 3 for m, you're going to get different numbers for all of those expressions. So if you take whatever value your m to z value is off your spectra, you can set that equal to m plus 1 over 1, and you can solve for m. Hopefully you guys know how to multiply by 1 and then subtract 1. Like, it's math. It works, I promise. There's a lot of practice problems like that in the book if you guys want to give them a try. I definitely recommend it because ESI is super cool, and I think it's really fun. I'm going to be working with it a lot this summer and really excited, but that's completely beside the point. But this is a topic we spent a good bit of time on in lecture like 2 months ago, and so I wanted to make sure it got brought back up just in case you guys saw it again. Sound good? Any other questions about it? Okay. All right. Higher order structure. So we talked about the 4 levels of structure, but then Dr. Hashim also gave us 2 very specific examples in class of how other things were structured. What were those 2 examples? Anybody want to take a guess? Ian. Keratin and collagen. It's kind of why I put them up here. Your guys' exam is multiple choice, so questions like, what's the structure of collagen, are really easy to ask in multiple choice. So I wanted to make sure you guys remembered to take a look at this. That's kind of the point of this whole presentation, by the way. You guys are going to be able to have access to this. You can take this with you when you go to study all of the crazy hard details that are going to be in the book. This is a general overview. You can look more in depth later. We've got collagen and we've got keratin. And there's a lot in the notes about each of these compounds. But what was the main structure of keratin? Did anybody remember before I put it up on the board? Kind of, maybe. We talked about it a lot. If you don't remember, it's right there on the projector screen right next to me. We've got the coiled coil. So we've got a set of alpha helixes that are coiled around each other. And then those coils of coils are coiled around each other. And then those are bunched together in different fibers. There's a really great diagram of that in the textbook. So if any of you have your textbook and want to pull it up, that'd be cool. If not, I don't have time to switch everything over, so I'm going to go ahead and leave that for your imagination. But we can make our keratin by forming the coiled coils and then stringing them together. For the collagen, it's a little bit different. And she mentioned this one specifically because of the weird twist it had. And so with this one, really what you need to remember is that you've got the 3 helixes that are coiled around each other left-handed, and then those units are coiled around each other right-handed. So they go one way and then they go the other way, and that holds them together a lot. Where do we see collagen in the body? Connective tissues, right? Also, fingernails. Fingernails are collagen. That's why they have you take collagen supplements and stuff when you're pregnant, because you can use that up and it makes your fingernails look really, really awesome. So that's cool. Where do we find keratin in the body? Hair, right? I don't think she specifically mentioned those, but it's good to know just for purposes of knowing things. And we get to the fun stuff. So I'm going to take a good bit of time for the myoglobin and hemoglobin stuff because I'm pretty sure this is going to be a question. I don't know what's going to be on your exam, but we talked about this for a week straight in lecture and 2 weeks straight in SI. So we want to go over this. So what are we looking at here? Anybody know? Right, the binding curve for which one? Myoglobin and oxygen, right. So in this case, we've got the chemical reaction that's being catalyzed by myoglobin, which is oxygen being transported through the body. So we're going to have essentially our protein by itself, myoglobin. We're going to have the oxygen by itself, and then we're going to have the form of myoglobin with oxygen bound to it. And that comes out in a reaction that looks kind of like this, and I'm going to write it up here because I can. I know it's not up yet, you can't see it, but we'll get there. Okay, can you guys all see that? Okay, I'm going to make it a little bit bigger. Fun stuff, okay. So we have this equation. And just like with any chemical reaction, well, not any chemical reaction, but most chemical reactions, this one's in equilibrium, hopefully. Because if all your body is ever doing is binding oxygen, you're not releasing any, then you have a problem. And if all you're ever doing is releasing oxygen, you're not binding any, you have another problem. So hopefully we're in equilibrium here. And when we look at equilibrium, what's the first thing that comes to mind when someone says equilibrium? Anybody? Maybe no one's just as big of a dork as me, but... KEQ, yes, equilibrium constants. They're important. They relate everything. So how would we write the KEQ of the forward reaction? What is it? Products of a reactants, right. In the body, are we more concerned about binding oxygen in the lungs or releasing the oxygen where we need it to go? Releasing it, right. Making sure that we have the oxygen being carried until it gets to our muscles is pretty important. And so when they do all of these hill plot kinetics, they actually look at the K equilibrium of the reverse reaction. So the dissociation, that should be MB. So it's the K equilibrium of the reverse reaction. And if the reverse reaction is going from MbO2 to myoglobin and just oxygen, which one's the products? Right, oxygen and myoglobin. And then the reactant is just MbO2. So we have our KEQ, which I think in the book it's also labeled as KD because it's dissociation. We have the myoglobin oxygen complex disassociating into just myoglobin and just oxygen. And now we can do all of the fancy math. So essentially what they try to do is they try to figure out like the best version. I'm going to go back to this plot for a minute. So when we report how much oxygen is in the body, we report it, instead of its concentration, oxygen's a gas, so figuring out the concentration of the gas is really confusing and a hard concept to like visualize and stuff. So instead they use partial pressure. And that's why we get the PO2 and stuff like that, is because we're looking at the partial pressure of oxygen in the body, because oxygen's a gas and not a solid that's being dissolved. And so for the YO2, does anybody remember what the YO2 represents? The percentage bound, right. It's the fractional saturation, I think they call it. But it's the fraction of what's bound over the total amount of protein that we have. And I'm going to write this on here, and it'll show up when I switch back to the DocCam. YO2 equals bound over total. So we have a percent here. We have the percent of the oxygen that's bound over the total amount of protein we have. Which one of the components of our reaction, I guess I'll switch over now. Which one of the components of our reaction represents the bound oxygen? MbO2, right. It's the one where oxygen is already connected to the myoglobin. So bound is MbO2. How many terms do we have total that involve myoglobin? In our reaction, how many different forms of myoglobin do we have? I heard 2. There's a bound and an unbound, exactly. So if we're looking for the total concentration, we want the total amount that's not bound to anything plus the total amount that's bound to stuff. Is this equation starting to look a little bit familiar? Cool. Okay. So we bind our oxygen, we get there. And how many oxygen can myoglobin bind? How many oxygen molecules? 1, right, exactly. So as far as this chemical reaction goes, all of our coefficients are 1. Does anybody remember from back in Gen Chem where coefficients go in our equilibrium equation? Superscripts, right, they're exponents. So in this case, the K equilibrium, these exponents, or these concentrations would all be raised to the whatever coefficients in front of them. In this case, those coefficients are 1, so we're going to ignore those. But I want to mention that because it's going to become important when we talk about hemoglobin. So those all have 1s as exponents there. Now we have these 2 equations. Let's turn them into something useful. So we've got 3 different terms in our YO2 equation. How can we get rid of one of them? I saw a hand gesture. I think you mean substitute stuff in. I'm going to go with that because it's right. Okay, so we're going to substitute this equation in here somewhere. We need to substitute it in somewhere useful. So how are we going to do it? What are we going to solve for? MbO2? Why? Because it's the easiest to solve for? What do you guys think? What occurs in our equation most often? In our YO2 equation? MbO2. It's on the top and the bottom, and that's not convenient. And we like things to be convenient because we're lazy scientists and we want math to be easy. So I'm hoping that you guys can all rearrange this equation. If not, this is how you do it. Okay. So now, let's plug that into our YO2 equation. Okay. So we know what our MbO2 is represented by. And this is going to be a crazy fraction, but just bear with me for a second. So we solve for MbO2. We still have myoglobin. and we solve for MbO2. MbO2 over KQ. So now what? Where do we go now? Take out MB from each term? Okay, cool. Let's do that. Oops, sorry. So let's divide out myoglobin, myoglobin, and myoglobin. I'll do this way for consistency's sake. And we left 1 plus O2 over KQ. Now what? This is still like a complicated, crazy fraction. What else can we take out from the bottom, or from the top and bottom? O2 over KQ? What if we just did 1 over KQ? Let's try that first. And I'm going to do that on purpose. Because do you guys remember the fractional saturation equation we looked at before? Oh, they actually don't have, I don't have it on there. Never mind. But let's pull out just the 1 over KEQ. If we do that, that leaves us with concentration of oxygen on the top. Oh, sorry. I don't know if I can, but I can do this. Technology. Concentration of oxygen on the top. We factored out 1 over KEQ from the top and bottom. Sorry, I wrote my fraction weird. But we're going to take out this from the top and bottom. So that leaves concentration of oxygen on the top. 1 divided by 1 over KEQ, and it's just going to leave KEQ. And then plus concentration of oxygen on the bottom. This is looking good so far. We're getting closer to what our YO2 equation from our notes in our textbook from a long time ago that we might not remember now, but we're getting close to what it looks like, so let's keep going. How did I say we represent concentrations of gases? I said it like 10 minutes ago. Can we measure specifically the concentration of a gas dissolved in something? Partial pressure, right. It's much easier to gauge the pressure. So let's fix that part of our equation. Now is this starting to look familiar? So far, so good? Okay. I'm hoping this is triggering your memory as we go through it, that you'll remember it. I meant to drop that pencil. Yeah. Okay. So now we've got partial pressure of oxygen over the equilibrium constant times partial pressure of oxygen. Except this is kind of really inconvenient because it's really hard to measure the equilibrium constant. So what scientists decided to do, and what we're going to prove works, is they said, let's make KEQ equal to a value that we can conceptualize, make it useful. Does anybody remember what the second term in the denominator of the actual hill plot equation is? It's like a weird constant that changes for myoglobin and hemoglobin. Kind of, you're on the right track. So this is a constant that is specific to whatever enzyme we're looking at. In this case, myoglobin. We're going to look for the partial pressure of oxygen required to make the saturation 50%. Conveniently, they set that value equal to KEQ, and this is why. So if we want our fractional saturation to be 1/2, right? That makes sense to everybody? Let's say we want YO2 to equal 1/2. And up here, let me zoom out actually. Wrong way. We said that YO2 was the bound over the unbound. And so at equilibrium, where the equilibrium constant is useful, we're going to say that the KEQ is equal to where it's half saturated. And where it's half saturated is where your concentration of unbound myoglobin equals the concentration of bound myoglobin, right? Half saturated, half of everything is bound. So if we have 1/2 bound over half unbound plus half bound, half over 1 equals a half. It's kind of like 1 over 1 plus 1, same kind of concept. Simplifies to 1/2. So whatever concentration of oxygen gives us a YO2 of 1/2, that's what they call the equilibrium constant. And that's why they do it that way, is because we can then cancel out, or sorry, this should be PO2, that's right. Whatever the concentration of oxygen that gives us a half saturation, they call that value the P50 value. I actually think it's written as subscripts maybe, or PO50, however it's written in the book. So we now have our P50 value, and if that's going to be equal to our equilibrium concentrations, then we're going to plug that in. We're going to plug that in right here. And that gives us our fractional saturation equation of pressure of oxygen, P50 plus pressure of oxygen. Sound good? Does anybody have any questions on this? On like how it's derived and stuff? Okay, cool. So now let's look at this graph that we had. We have this equation that we created for our YO2, and now we're going to put it on a graph so that we can look at it. So our constant is P50. That's not going to change. Our variable, our x variable, is going to be the pressure of oxygen because we can control that. We can put more oxygen into our systems. We can take more oxygen out of our systems. Please keep breathing. That'll help a lot with that process. And so once we breathe and we get more oxygen in, we take more oxygen out, then we can calculate the YO2, the saturation of how much oxygen is bound to myoglobin. And you get a fancy plot that looks like this. And they have so wonderfully marked here the P50 value, where the saturation is 50%, whatever concentration that is of oxygen, that's our equilibrium constant in this case. And for myoglobin, it's like 3.26 or something like that. They give you that in the book. I don't think Dr. Hashim is making you like memorize it. That's usually given to you, so you don't have to worry about it. So everybody cool with the myoglobin part of this? Sweet. Okay. So now we have hemoglobin. And hemoglobin works a lot like oxygen, or myoglobin. What's the main difference between hemoglobin and oxyglobin? Hemoglobin and myoglobin is what I meant to say. Right. Hemoglobin is cooperative binding. What does that mean? Okay. Exactly. So what Ian is saying is that with hemoglobin, because we have multiple binding sites, we have multiple oxygen molecules that can go to one specific hemoglobin molecule. And when you bind one oxygen, it's then easier to bind the next one and then easier to bind the next one after that. And so in this case, we have to deal with those coefficients I was talking about earlier because the coefficients of our chemical reaction for hemoglobin are no longer 1. It's still one hemoglobin molecule, but there's multiple oxygen molecules that can be bound, and so we have to take that into consideration. And that's where we get these Hill constants. So when we look at this equation here, this YO2 equation looks pretty similar to the one we were just talking about, right? We've got that in here. The only thing that's different is that n exponent, and that's going to normally be a value for hemoglobin between like 2.8 and 3.4, I think, are the limits. It's going to probably be given to you if you have to see it. And that's just representative of the value of the cooperation. We're hopefully going to have time at the end for questions too. So if you think of any questions between now and then about hemoglobin and stuff, we can talk about it too. Okay. So this is a little bit about hemoglobin and the different binding sites. So we have two forms of hemoglobin. What are they? T and R, right. It's right there on the board. See, I keep asking you these questions to see if you're paying attention to what's on my projector. It works. So we've got T and R. We've got, which one's active? R. Which one's inactive? Okay. I say active, inactive, bound, unbound, same kind of process. So when it's bound, we've got the R form, right? Yeah. Bound is R form, T is unbound. And when we change where the oxygen is bound, or when the oxygen is bound to it, we're changing the conformation of our protein. So it's still hemoglobin. It's still that same polypeptide sequence. It just changes its shape and does different things. So where does oxygen bind to hemoglobin? To iron. Okay. What is the part with the iron called? It's a binding group, right? There's a specific name. Anybody? I say this because your test is multiple choice and definitions are going to probably be a thing. Does the term heme group ring a bell? Oh, is that what you said? Oh, I'm sorry. I'm like half deaf. So there's that. So it is the heme group, and great job. Thank you, Alex. So we've got the heme group, also known as the prosthetic group. It's this extra bit of not protein material that's stuck to our protein and does the stuff we need it to do. So in this case, our binding site for hemoglobin is going to be this prosthetic group. And it's got some iron in the middle, and it's going to stick the oxygen right in the center. And based on when the oxygen's bound, the heme group looks a little different, which makes the protein look a little different. And we get these conformational changes we talked about going from the T to the R state. Sound good? Cool. And I think specifically, she's not going to make you remember, like, it's the F8 helix or whatever. I don't think she made you do that for the last test, so I can't imagine she'll make you do it for the final. This is a little bit about the respiration cycle. Why do we need hemoglobin in our lives? Deliver oxygen to the muscles. We kind of need it for breathing. So it makes sense that the whole respiratory cycle is kind of important for hemoglobin. So in this case, we're going to start in the bottom left here with the inhale section. So we're going to bring air into our lungs, and that free oxygen is going to bind to the hemoglobin that's in the blood vessels in the lungs. And you can breathe out now. It's going to be okay. So then that's going to be transferred over through the bloodstream to the muscles, and the hemoglobin is going to release its oxygen. It's going to do its stuff in the muscles with the myoglobin. The muscles are going to work. We're going to move and not be dead and stuff. And then eventually we're going to use that oxygen, go through respiration. We're going to form carbon dioxide, and then that carbon dioxide is going to be carried out of the system by carbonic anhydrase and go back out to our lungs so that we can exhale and get rid of the carbon dioxide and not suffocate. The Bohr effect was a really weird topic that Dr. Hashim brought up that a lot of people were confused on too. So I wanted to also mention it here so that we can clarify that. Does anybody know what the Bohr effect is? The thing, yeah, it's a thing. A little bit. So the Bohr effect has a lot to do with pH and stuff. Amami, did you want to add to that? Right. So with the Bohr effect, we're going to be dealing with the carbonic anhydrase reaction and the hemoglobin reaction. When we take up oxygen, we're going to be a little bit changing the pH of our system because of the conformational changes in how the oxygen is bound. So the reaction mechanism for carbonic anhydrase that we're going to go through in a minute, that's going to release a proton, and that's going to make the solution, our body, more acidic. And then we can use that proton that's released, the hemoglobin can use it which makes the binding site for oxygen have a higher affinity. And so when we retake up that proton, we're decreasing the pH again, which increases the affinity for hemoglobin. And that's what the Bohr effect is talking about. It's how the pH affects the changes, or affects the activity of hemoglobin, if it makes it better or worse. And so if you look at this diagram, it's a little bit confusing, but that's why I brought it up here so we could go through it. So we inhale, we bind oxygen and hemoglobin. We carry it to the muscles, we release carbon dioxide, and then we start the carbonic anhydrase. When we use carbonic anhydrase, we release a proton, which increases the acidity, so it decreases the pH. When we use that proton with hemoglobin, we're decreasing the H+ concentration, and so we're increasing the pH. And our hemoglobin is a lot more readily available, I guess, for binding oxygen. So we're going to lose the proton, which is going to it's going to decrease the pH again. We're going to bind oxygen. We're going to start this process over. Does that make sense? Right. So the more protons that are off because the hemoglobin is going to take up that proton, and it's got to release that proton again before it can take up oxygen. So once we release the proton, that's going to make our bloodstream more acidic, so it's going to have a lower pH. So decreasing the pH is going to have the effect of making our hemoglobin more susceptible to binding oxygen. So it's going to be better for the hemoglobin. Make sense? Are we good? And then the opposite is true for going the other direction. So she asked a question about Bohr effect. You think you got it now? Okay, if you don't got it, there's a figure in the book, but we got to keep going because we have less than an hour in all of Michaelis-Menten. So it's going to be super fun. This is the carbonic anhydrase model. I know she talked a lot about catalytic mechanisms way back when, chapter 13, I think. This was one of the ones she specifically mentioned, was carbonic anhydrase. There was the RNase and the serine protease mechanisms, and I've got examples of all of these. But in the carbonic anhydrase, what's the main amino acid we're dealing with here? Which amino acid is that? Histidine, right, yeah. So as you can see, we've got the zinc atom that's coordinating, it's being coordinated by the 3 histidines. And we're going to attack our carbon dioxide with water, and protons get transferred around. So what kind of catalytic mechanism is this? Does anybody remember the types? Transferase? This is a more general category. There were 5 types of catalytic mechanisms, and I know I have them listed somewhere else in here. What kind of chemistry is happening when we're transferring protons? Acid-base, right. So in this case, we're moving a proton around, and so acid-base chemistry. That's our reaction mechanism for carbonic anhydrase. There's a full-scale one in the textbook. Where did I have the list? Here's our mechanisms. I had them somewhere. Acid-base, covalent, proximity and orientation, metal ion catalysis, preferential transition state binding. All of those types of mechanisms appear in the body somewhere. In the case of the specific ones that Dr. Hashim wanted us to remember, I think most of them were acid-base because that's like the most common one in the body. So we'll skip this later. I meant to have this earlier in the presentation and I forgot. Okay, so we talked a little bit about enzymes and enzyme catalysis and stuff. So do you guys remember rate law stuff from Gen Chem? I know that was probably a long time ago. It was for me. Yeah, a long time ago. All of the Michaelis-Menten stuff comes back to Gen Chem's rate law. That's why I put this in here. So with the rate law, that's where we get those different Ks for Michaelis-Menten. We'll go through all of the Michaelis-Menten stuff in a minute. I know Dr. Hashim published in her notes the entire derivation of Michaelis-Menten. So I'm going to let you guys look through that on your own time because, again, time constraints. That's in the Module 2 lectures. Somewhere in there. She's got exactly how to do Michaelis-Menten, exactly how she wants you to do Michaelis-Menten, and it's very detailed in her explanation for it. But those Ks and like big Ks and little Ks, they come back to this rate law. The rate law is where the little Ks come from. So that's the velocity, I guess, of the reaction going forward, and the different reactions I have on another slide in the future. But I would definitely, if you're not sure on that, go back to your Gen Chem notes and review the rate law stuff. I guess I have Michaela's Menten coming up in a minute, so I'm going to save that for a minute. But the enzymes specifically function to bind substrates and make our reactions go faster and require less energy to be input to break things down. That's why we have enzymes, and that's why they're super important. So we have to know about them. This table in the textbook, table 11.2, is the different specific types of enzymes, which the enzyme types, I want to point out, are different than the reaction mechanism types. So if she gives you an example of an enzyme mechanism of like the catalytic triad and showing you how like the different molecules interact and stuff, and asks you to pick like what kind of enzyme is this, that's when you want to go to this list, table 11.2 in your book. The oxidoreductases do oxidation reduction. I think transferases, they transfer functional groups. So if you move like a carboxylic acid group from one molecule to another, that would be a transferase. Same for hydrolases, lysases, liases, isomerases, and ligases. Those are all specifically for those types of enzymes being catalyzed, or enzymes that are being used for catalysis. So if she gives you a mechanism and says what kind of mechanism this is, that's a different question from the enzyme classification. So we've got what kind of enzyme, what kind of mechanism, and the what kind of mechanism comes back to our list of acid-base, covalent, proximity and orientation, metal-ion catalysis, and transition state binding. There's a lot of explanations of this in the book, but I know that was a confusing question on one of her past exams, so I wanted to mention that again. Sound good? Okay. So a little bit more about enzymes. We've already talked about serine proteases today. Chymotrypsin, trypsin, elastase are the ones that Dr. Hashim wanted us to remember. The specific enzymes, different enzymes work in different ways. So a lot of enzymes use cofactors, things that aren't proteins, to help them with their catalysis. So what's an example of a metal ion cofactor that we've seen before? Iron for hemoglobin. Kind of, yes and no. It falls under that category, but it falls under a different category on this chart, so we'll go into that one in a minute. Does anybody remember any others? Zinc, yes. There's like a specific one with zinc. I'm doing this on purpose. Zinc fingers, yes, that was a thing. Zinc fingers use zinc ions as their cofactors specifically for the reactions that they catalyze. Coenzymes are a little bit different. So coenzymes are other groups that are made of amino acids that are attached to our enzyme itself. And under coenzymes, we have two categories: co-substrates and prosthetic groups. Co-substrates we didn't really talk a lot about in class. We didn't talk a lot about like allosterism or anything like that. But what's an example of a prosthetic group that we've seen? This is the one we were talking about a minute ago that I said was in a different category. Iron and hemoglobin, and the heme group in hemoglobin, that's the prosthetic group. That's the important part to remember there. So if she asks, where do we find an example of a prosthetic group, we can say hemoglobin, everybody's happy. Okay. Talked about the catalytic mechanisms. This is the specific mechanism for the acid-base catalysis. I don't think, because you guys, it's multiple choice, I don't think you're going to have to like draw anything, but if she asks you to identify certain parts, this is definitely going to be good to know. So obviously RNase A is one form of an acid-base catalysis. Does anybody know of two other examples of enzymes that use the acid-base mechanism? We've talked about one of them today. We've talked about two of them today, actually. I'm going to go with, you want me to tell you because time's up. Okay, so the other two that we saw, we use RNase A. We also use serine proteases, so our trypsin, chymotrypsin, elastase, use an acid-base mechanism. We're transferring a proton around from our substrate back to our catalyst and around the amino acids and around. So if you follow this reaction, You can see in the beginning, we attack and we steal a proton. And then the proton is no longer attached to the serine because the serine is attached to our substrate. Then the proton gets transferred back to the amino group, back to histidine, back to the proton, or back to the substrate, back to the protein again. So if you follow the mechanism, the main changes here are nucleophilic attack and transfer of a proton. Transferring a proton, acid-base catalysis, boom, everybody's happy. So if you're asked for an example of an acid-base mechanism, serine protease is one that you can think of. Another one we talked about, oh, that's not one. Go back, okay. Not the example I thought was coming up next, but lysozymes, catalytic mechanisms, what are lysozymes used for? Right, they break down the bacterial cell wall. What's the bacterial cell wall made of? What is it? Peptidoglycans, right. So we've got peptides that are attached to glycans, which are the carbohydrate forms. Where does specifically lysozyme cleave? Does anybody remember the acronym NAGNAM? I just thought it was funny because when I draw this mechanism, it's like it splits between NAGNAM. So if you remember that, you can remember this one because it's the N-acetyl muraminic acid and the N-acetyl glucuronic acid or something like that. I forget what it stands for. I just remember NAGNAM or NAMNAG. And that big polymer chain is cut between the two monosaccharides, just like is shown in this mechanism. And again, I don't think she's going to make you like memorize this one exactly, but it's important to know how these mechanisms work and how to read them. So if we start with binding the substrate, we have this binding pocket, which is, what amino acid is this? It's labeled. Can you see it? You may not be able to see it. Glutamine, right. So CH2, CH2, and the carboxylic acid is glutamine. And then asparagine, CH2, and the carboxylic acid. Then we bind our substrate, comes into the binding pocket, and our glycosidic bond attacks we steal a proton from our enzyme. Proton transfer, what kind of mechanism is this? What did we say earlier? Acid-base, right. So we transfer protons around, back and forth from water to the substrate to the enzyme and back. We've got an acid-base mechanism, all of how the lysozyme breaks down N-acetyl muraminic acid from N-acetylglucuronate. Sound good? Cool. N-acetylglucopyranose is what I'm thinking of. Okay. Everybody's best friend, Michaelis-Menten. How many of you remember Michaelis-Menten from start to finish? Yeah? Start to finish? Okay. Well, start to halfway and 3/4 to end, yeah. That's probably, given that it's been so long since Michaelis-Menten, that's probably average. That's okay. I personally have to like sit through it and think it out and stuff, so don't feel bad if you can't like spit it out off the top of your head. It's a long process, and we don't have time for it all now, but the important part of Michaelis-Menten that I want to cover is the conceptual part. So the math is all explained in Dr. Hashim's notes. You can go back to those, and they're super awesome. The conceptual part is what I want to focus on here. Why do we talk about Michaelis-Menten kinetics? Okay, yeah, so it has a lot to do with the substrate binding to enzymes. What specifically are we throwing into the mix when we're talking about Michaelis-Menten? Concentrations of the substrate and what else? And the enzyme and the complex together. What else is added in, or can be added in some cases? Anybody remember? What? What is it? Sorry? The products, okay, kind of. It's the inhibitors. We can throw in some inhibitors into this mix, and that's going to make our Michaelis-Menten kinetic equations look crazy, but it's doable because if we add inhibitors, we're changing the rates of reactions with our enzymes. So let's say we have this reaction, and it's kind of okay. It goes all right by itself. We add an enzyme, and it goes 10 times faster. This is awesome for us. We have an enzyme. We're not wasting as much time in this reaction. It doesn't require as much energy. This is great. But let's say this enzyme has a specific inhibitor, and that inhibitor is found in Coca-Cola. And I drink Coca-Cola like most people drink water. And so I drink a lot of Coca-Cola. I now have a lot of inhibitors in my system that's going to block that enzyme. And so instead of being 10 times faster, maybe that enzyme's like helping a little bit. So like some of that enzyme's not inhibited, but most of the enzyme gets inhibited, my rate of reaction is going to go back down. And that's the same concept that we're talking about with inhibition with Michaelis-Menten, except it's not really caused by Coca-Cola. It's caused by whatever other inhibitors you put into your system. And so when we talk about different types of inhibition, we've got the competitive inhibition, where you've got your Vmax, which is the maximum velocity of your reaction. The speed of the reaction when you put in a catalyst, that's not going to change. That's going to stay the same. But when we add an inhibitor, we're going to change what the equilibrium concentration of that reaction is going to be. Does anybody remember this from class, hopefully? Is this ringing any bells? Okay. So based on this graph, and I know that some of the words might be hard to see, if we increase the inhibitor concentration, so we go from the flatter line to the steeper line, how is Km apparent affected? Does the Km increase or decrease from the one with no inhibition? Increases? Right. Because we're going to increase our concentration of inhibitor. Everything else stays the same. So when there's no inhibitor, we have a Km that we found from the slope. And as it's listed on the slide there, the slope is the alpha value times the Km divided by Vmax. We're not changing Vmax in competitive inhibition. We can see that by the fact that our y-intercept doesn't change. If we're not changing Vmax, the only thing that's going to change if we change the inhibitor concentration is the Km for competitive inhibition. And if the Km changes and our slope changes, we can use the change in the slope to figure out the change in the Km because they're directly proportional. So if the slope increases, that means our Km also had to increase. And that's the trend we see for competitive inhibition. Don't worry, there's a table at the end of this. It'll all make sense. This is the reaction scheme, by the way, for competitive inhibition. So what? Km is going to be the equilibrium constant of this overall reaction, of the substrate going to product with the enzyme complex in the middle. And so to find that, we have to consider all of these possible reactions that can take place. And that's where the little k's come in with our rate constants and everything from the rate law from Gen Chem. We have to figure out the rate of the forward reaction, how fast we're making the enzyme substrate complex. We've got to consider the rate of the reverse reaction, how fast we're destroying the enzyme substrate complex and going back to our reactants. And then the rate of our second reaction, how fast we're forming product and releasing it from the enzyme. With inhibition, we also have to consider, with competitive inhibition, how fast we're destroying the enzyme in the beginning to where there's not going to be any more reaction happening. So the competitive inhibition is forming the inhibited enzyme at the very start of this reaction so it can't take place. That's a different case than our uncompetitive inhibition. Uncompetitive, we're going to inhibit that ES complex. So we're going to bind the substrate, and then we're going to bind the inhibitor, and nothing else can happen. There's not going to be a reaction to form the products. So in this case, our Km's not going to be affected, let me think about this for a second. I have to cheat and go to my table. I don't want to tell you guys the wrong trend, because that's what she's going to ask you, though, I'm sure. Okay. So our Km is going to be affected, and our Vmax is going to be affected. So that's not true. Our slope doesn't change. Slope doesn't change, Km doesn't change. You can tell by this graph here, all those lines are parallel. Everything's cool. With uncompetitive inhibition, we're changing the maximum velocity of our reaction. If we inhibit this reaction somewhere in the middle, then it's going to start, but the overall rate to completion is going to go down because we're cutting it off halfway on some of them. Does that make sense? That's kind of a simplified conceptual analogy. It's not really cutting it directly in half for some of them and not for others, but that kind of works for explaining it. And so in this case, our Vmax is going to change, and that's going to be our y-intercept. So if our y-intercept is 1 over Vmax, which it's labeled on the board here, and you can look at all the fancy math she explains in her PowerPoints to show that that is how that works. If our intercept is alpha prime over Vmax and alpha prime is constant, what happens when we increase the concentration of our inhibitor? What happens to the y-intercept of each of these lines when we increase the concentration of the inhibitor? The intercept goes up, that's right. So the intercept increases. So is Vmax increasing or decreasing? Vmax is decreasing, right, because the y-intercept is alpha prime divided by Vmax, inversely proportional. When one goes up, the other goes down. So if the intercept is going up, the Vmax is going down, so when we have uncompetitive inhibition, increasing the concentration is going to decrease our reaction velocity. Yeah. Yeah. Right. So the question is, was this from, which was this from? An old exam. Okay, so this was a question from exam 2, and it says, it gives you the inhibitor concentrations, the Km apparent, and the Vmax. And the Km goes without the inhibitor as 0.02, with the inhibitor as 0.01. Not really a significant change. There's probably some statistics involved to prove that. But the Vmax goes from 150 to 90. So with a drastic decrease in the Vmax and a relatively constant Km, we're going to have the uncompetitive inhibition because Km is staying the same and Vmax is decreasing. Vmax is decreasing. Make sense? Cool. All right. So that's the uncompetitive. And then we have the combination of the two. So because nothing in the human body is simple, we have some cases where, like in our competitive inhibition, we block the enzyme right from the start. And then we have some cases, like in uncompetitive inhibition, where we block the enzyme substrate complex. And then, of course, because this is biochemistry and it's not easy, we have some where it does both. And there has to be math to account for that. And so we have this strange-looking reaction that is inhibited in 2 different places. And we have this strange-looking graph where it's not parallel and it's not the same intercept anymore. How do we account for this? We look at the math. The math is all laid out in the textbook. It's all there for you. I don't have time to walk you through it step by step. But let's look at the same trend. When we have mixed inhibition, how does the Vmax change when we increase the concentration of the inhibitor? We're increasing the concentration of the inhibitor, and so we want to know how the Vmax changes. Do you think it increases? I've got a vote for increases and a vote for decreases. It's actually decreasing. So if we look, the y-intercept is still found the same way. We've got the alpha, which in this case is just a proportionality constant, divided by the Vmax. And so when we increased our concentration of an inhibitor, our y-intercept increased. And since it's inversely proportional to the Vmax, that means Vmax has to decrease. When you put a larger number in the denominator, you get a smaller number out. Same concept. So we're going to decrease our Vmax with mixed inhibition. What about the Km? The Km increases? Okay, why so? This is the hard part to explain and visualize and stuff. So we can still find the Km from the slope. The slope is going to be our proportionality constant, which is alpha. And alpha is actually described on the slide as 1 plus the concentration of the inhibitor over Ki, which is the equilibrium constant of the inhibition parts of this reaction. Ki is going to be a constant. It's not going to change. So if we increase the value of, or if we increase the concentration of the inhibitor, how does that change alpha? As listed on the equation up here. Alpha is going to increase, right? If alpha increases, And our slope we see can increased. We know our Vmax was decreasing. So can we You guys are all staring at me or staring at the screen like you hate everything. The answer is no, we can't make a prediction on that. It'd be way too hard. We've got different variables moving in different directions all over the place, and that brings me to this table that's going to lay out all of our trends. So we have, like we said at the beginning, first of all, we can have no inhibition, which would make our lives super easy and super great. We could have competitive inhibition, where we're only going to increase the Km apparent. That's the only thing that changed. We could have uncompetitive inhibition, where we decrease the Km and decrease the Vmax. Or we can have the mixed version, where we definitely are decreasing Vmax because our y-intercept is going up. And the Km might go up or might go down. It totally depends on the conditions of our reaction that we're looking at. Sound good? This is the table that's going to be super important. It's going to be your best friend, table 12.2 from the textbook. If you can remember these trends, I think even when we did the exam with us on it, exam 2, I think she even said just be able for the inhibition parts to understand how the trends work. Do what? Let's go back and look at it. So we said for uncompetitive, we're looking at the change in Km. I might have misspoke. So let's look at how we can find Km. The slope is Km over Vmax. And in each of these situations, we're changing Vmax, right? We can also find Km by looking at the x-intercept. And I meant to bring this up when we were talking about it. Thank you for reminding me. We can look at that x-intercept. That x-intercept is our proportionality constant divided by the Km. Now that proportionality constant is listed on that equation that's on the slide in the chart as 1 plus concentration of the inhibitor divided by the reaction or the equilibrium constant for the inhibition reaction. If we increase the concentration of the inhibitor, what happens to alpha prime? Ki is staying the same. 1 is just a constant. So what happens to alpha prime as on the board when we increase the inhibitor concentration? It's going to increase, right? Because we're going to just be adding larger numbers to 1. So if we have our proportionality constant is increasing, it's still 1 point something times, or 1 point something, because we're adding a fraction to 1. That's based on the equation that we're given. And then our x-intercept is going to be how we can find our Km. So in this case, we're going to take our x-intercept and forget that it's negative for a second. So all of those values, if we got the specific values, we turn them into positive numbers, and that value is equal to alpha prime over Km. If that value gets bigger, the absolute value, I guess, if that value gets bigger, how is Km changing? We know that alpha prime is getting bigger. How does Km change? It's a trick question. The answer is it depends on the conditions of the reaction. But because of, and this is all in the explanation in the textbook that I don't have time to go through step by step, and I wish I did, because it's a really confusing topic. But with the trend specifically, most of these reactions, they're going to show that decrease in Km. And it has a lot to do with the conditions, and it has a lot to do with how the inhibition is affecting the enzyme substrate complex. So if you take a look through the textbook's explanation of that, it has a really detailed and really great explanation for that in chapter 12. I know that probably didn't really answer your question, but again, for Dr. Hashim's exams, I know for exam 2, she said specifically remember the trends, and this is the table that she pulled the trends from. So if we know this table, then we're good for life, and everybody's going to be really happy, hopefully. Sound good? Okay. What time is it? 8:05. Okay. Pause for the cause. I need to find out how much we have left here. I'm going to try to get through the rest of the PowerPoint. We only have about 15-ish more minutes before we got to wrap up to get ready for the next session. But I wanted to talk about, we just talked all about proteins and enzyme kinetics and how all of the protein stuff that we need to know. But there's 3 other giant classes of macromolecules we need to know for biochem. So let's go through those real quick. So we start with carbohydrates. What are the functions of carbohydrates? What? They store energy source, and they structure materials, and they're signaling with cell receptors, and they form the structural components of nucleic acids, which is super important. If we didn't have sugar, we don't have DNA, and if we don't have DNA, we don't have people. And I like being a person, so like that's kind of beneficial for us. We really need sugars in our lives. Carbohydrates, we have the different types, aldoses and ketoses, alpha, beta, L, and D. And hopefully you guys know how to go from the Fisher projection to the cyclic. But as was brought up in one of my, I think it was probably my first review, there's a really great mnemonic for remembering how to go from Fisher to Haworth projections. Ian, what was it? Downright uplifting. So a play on words here. It's downright uplifting because it's really nice and awesome. But on a Fisher projection, everything that's on the right is going to be down on your Haworth, provided you draw your Haworth structure properly. So in this case, we're drawing the oxygen in the upper right of a 6-membered ring and in the top of a 5-membered ring. But in that case, everything that's on the right of your Fisher is going to go down, and everything that's on the left in your Fisher is going to go up. And it's super great until we get to that anomeric carbon and the reference carbon, and then things get a little weird. For what's the easy way to remember alpha and beta? Which one's same side, which one's different side? Beta is same side, and alpha is the other side, is opposite sides. So when in doubt, if you're in a rush, that's probably going to help you. It gets a little tricky with like fructopyranose and fructofuranose and like the ones that are a little bit harder. The other way to remember that is if it's on the same side, then it's beta. If you can't tell, like if there's something else attached to the anomeric carbon or something, you can assign the configurations. And so beta will have R and R or S and S for the anomeric carbon, and the reference carbon, which is the chiral carbon that's farthest from the anomeric carbon. So whichever one is, it still has to be chiral, and it's going to be whatever position is farthest away from the anomeric carbon. If both of those carbons have an R configuration, or both of them have an S configuration, then it's beta. If one is R and one is S, then it's alpha. Sound good? It's similar to the same side, different sides, because it's same configuration, different configuration. People just don't like to think of it that way because for some reason everybody's terrified of configurations. They're fun. It's great, I promise. Okay. And there's all sorts of stuff about polysaccharides. We put sugars together. We can use them for storage when we have glycogen and cellulose. We can use them for structures when we have chitin and chitin for exoskeletons and other different polysaccharides for that. And then glycosaminoglycans when we have different tissues like cartilage and tendons and stuff. We're going to modify our sugars and put them on like proteins and lipids and stuff so that we can form structures in the body. Sound good? Okay. So then we get to nucleic acids. There's a lot more on carbohydrates in chapter 8 and stuff, but I think mainly as long as you remember the specific functions and how they're all put together, then you should be fine with that. Nucleic acids, obviously, sugars play a part of this. What are the 3 components, of nucleic acids? There's the ribose or the deoxyribose, so the sugar, the base, the nitrogenous base, adenine, guanine, thymine, adenine, guanine, thymine, cytosine, or uracil. What else? And the phosphate. We got to have the sugar and the phosphate to make the backbone. We got to have the base to do everything else that we needed to do. And so we can do base pairs. This was the sequencing for DNA. They fragment it, and they run the dideoxy method. So do you guys remember how to do the dideoxy method for sequencing? She went over it in class a lot. Did she talk about it on Monday? Oh, on Monday? Okay, cool. So basically, we're going to run a reaction with 4 vials. And each vial is going to get everything you need for DNA synthesis. And then one of each of those vials is going to get a messed up nucleotide. And that messed up nucleotide is going to stop the synthesis wherever it gets attached. So in one flask we'll have our dideoxy-Cs, and one task we'll have our dideoxy-Ts, and so on and so forth. The ones that have dideoxy-Ts, sometimes they might incorporate a regular thymine. Sometimes they're going to incorporate the dideoxynucleotide, and it's going to stop the sequence or the synthesis right there. And then you can find the fragments, run a gel, and figure out the sequence from there. Question? Right. Then Right. So what they do is they take each of those flasks, they run the reactions to completion, everything else all set to go, and then in our gel, we have a column for C, a column for G, a column for T, and a column for A. And based on where the fragments come out, they're going to be sized in this order. We're going to see them as smallest fragment to largest fragment, and based on where they are in the gel, we can find out what the end nucleotide is, and then get the sequence going from bottom to top will be the complementary sequence of the strand we started with. This diagram is really great for explaining that as a visual. There's another really great explanation of it in the textbook. I think the internet. So I don't think it's in the textbook, but there are some great visuals in the textbook for it. Gel electrophoresis to separate fragments. We talked about that earlier with proteins. We have restriction sites that are going to cut either sticky ends or blunt ends. And I'm breezing through this because I have to wrap up soon. PCR is another. It's creating more DNA so we can do more analysis. It's really important if you're in forensics like me. And then we can move on to lipids real fast before we have to wrap up. So there's a lot of really great functions for lipids. Anybody know what they are? They're components of biological membranes. They store energy. All the ones that are listed on the board here. We have a lot of different kinds of lipids. We have like cholesterol. We have different steroids in our body. We also have like fatty acids and oils. We have glycerophospholipids, which are in our membrane bilayers. They've got two tails, and then they've got a phosphohead on the top. That's what makes our cell membranes. We have triacylglycerols, where we have a bunch of fatty acids attached to glycerol, and that's going to have a lot to do with the storage of energy and stuff in our body. So there's a lot of information about this in the third module of Dr. Hashim's notes, so you can look a lot of that up there. They're a lot controlled by hydrophobic effects because you have, for lipids, a lot of hydrophobic sites, essentially. Our fatty acids are giant chains of hydrocarbons. Nonpolar molecules don't like water, and so they're going to be directed a lot by where the water is in our systems. Same for our proteins. If we have a lot of hydrophobic chains, then they're not going to be outside in the cytoplasm, they're going to be turned in. Same kind of concept. Here's a picture of a membrane. It's got proteins in it. Super useful. I want to go forward a little bit to the transport stuff. There's different types of transport, active and passive. What's the difference between active and passive? One uses energy, one doesn't, right? Okay, so active is going to use energy. Passive is just going to let things go down the gradient from high to low. And there's, I think she mentions 4 different types of passive. The ionophores, the porins, the ion channels, and transport proteins. And she gave you guys like specific examples of each. So you can look up the specific mechanisms of those in the book or in her lecture notes. I think it's lecture 29 is the one with all of the passive transport stuff. And then lecture 30 talks about active transport. With the ATPases, there's like 5 different types. The ABC transporters and the ion gradients. Where with the ion gradients, a lot of people don't want to label this one as active transport because they forget that not all active transport requires ATP. In the case of the ion gradients, instead of using ATP, they use the energy from the gradient itself and the voltage gradient essentially to power their reaction to the cell. So there's a great example of the ion gradients in the book with lactose permease. I would highly recommend you looking through that section just so you familiarize yourself with the differences in passive and active transport. And that's going to wrap up everything for today. For Dr. Hashim's section, your exam is not just the material I talked about. I didn't talk about chapter 13 and 14 on purpose because, as you can see, we're already out of time. I would not have had time to go through it. But she is going to put 10 questions from the 4th module on the exam. So 30 questions will be from all the stuff we just talked about and anything else she covered in class. And then 10 questions will be on the last module. Does anybody have any last-minute questions for me? Okay. No problem. So we're going to have to wrap up. My throat's killing me. And it's somebody else's turn for this room. So you guys are set. I'm going to put this online so you guys can look through it and study from it. Good luck on Friday. If you're in Kazuli section, good luck tomorrow. And you all rock. Make sure you do your surveys, and I need those back. This is session 51. That part